Very good afternoon to all the EAPCET aspirants. This is your Prasanna Maam from NEET World, and here we are in our third chapter, that is photosynthesis in higher plants, from the most important unit, that is plant physiology. So first, with respect to this chapter, what you have to cover, you have to keep it in your mind, like what you have to cover, all the concepts that you have to cover from this chapter, you have to have a clear idea about this. Fine, right? All the concepts that you are going to cover from this chapter, just go through this. This is a common chapter both for your NEET and MCET. Fine, right? And what are the concepts that you have to cover here is the first and foremost chapter, the first and foremost concept is your early discoveries. Like, who said that these raw materials are required for the process of photosynthesis? What is the product of photosynthesis? How is that happening? Who discovered oxygen? Who discovered action spectra and absorption spectra? All the early discoveries that are given in the beginning of this chapter are important. Followed by action spectra and absorption spectra, the graphs which are given, both action spectra and absorption spectra are important. And then followed by photosynthetic pigments by looking into light harvesting complexes, followed by the organelle of the chloroplast, okay, followed by chloroplast organelle. After this, what you are going to learn? Chloroplast organelle, and then after learning the chloroplast organelle in detail, you have to move on to locations of light reaction and dark reaction, okay, locations where are the sites that light reactions and dark reactions are occurring inside the chloroplast, that you have to know. Locations or sites of both light reaction and dark reaction. Location and sites for light reaction and dark reaction, where are they occurring inside the chloroplast. And in detail in light reaction, you have to go through three sequential steps. One is absorption of light in the form of photons. Second will be photolysis of water. And third step of light reaction is formation of assimilatory power, that is formation of ATP and NADPH. Dark reactions, calling light independent reactions as dark reactions is a misnomer, but then this takes place both in the presence and absence of the light, but it is dependent on the products of light reaction, but not directly dependent on the sunlight. That is why these are addressed as light independent reaction. Three major cycles you have to know from light independent reaction: C3 cycle, C4 cycle and CAM cycle. This is how the glucose is fixed in different plants. C3 cycle is the common cycle in all the plants. C3, C4 and CAM cycle. C3 cycle occurs in all the plants, whereas C4 cycle occurs only in the tropical plants or C4 plants you call it as, whereas CAM cycle occurs in the desert plants only. Desert plants or plants belonging to arid regions. And lastly, factors affecting photosynthesis. What are the factors that are affecting photosynthesis? Factors affecting photosynthesis. Here itself, Blackman's law of limiting factor will come into picture. Okay, Blackman's law of limiting factor. Clear till here? So these are the concepts that you have to exercise from this chapter. Now, first, what are the raw materials of photosynthesis? How photosynthesis occurs? This we will learn by taking a standard equation of photosynthesis. One standard equation of photosynthesis. What is the standard equation of photosynthesis? See here. Okay. It is 6 CO2 plus 12H2O in the presence of sunlight, which I am taking in the form of photons, and chlorophyll. And what are the products formed at the end? Products formed at the end are glucose, C6H12O6 plus 6H2O plus 6O2. Here we go with the raw materials if you observe. First, 6CO2 and 12H2O. Who said that carbon dioxide is essential for photosynthesis? Here itself you take down the discoveries and I will tell you the experiments in detail in the coming slides. So, first, carbon dioxide is essential for the photosynthesis is said by Mole's half-leaf experiment. Mole's half-leaf experiment. Water and sunlight are essential for photosynthesis and oxygen is formed at the end of photosynthesis is said by Jan Ingenhouse. Okay, he is the scientist who said oxygen is liberated at the end of photosynthesis. Fine. Glucose is the product of photosynthesis is said by whom? Glucose is the product of photosynthesis is said by Julius von Sachs. Fine, right. Now, when it comes to oxygen, who discovered oxygen? Oxygen is discovered by Joseph Priestley. Joseph Priestley discovered oxygen. Fine. And who said that oxygen is coming from water molecule but not from carbon dioxide? Okay. Till most of the time the scientists felt that oxygen which is formed at the end of photosynthesis is coming from carbon dioxide but not from water molecule. Later it is the scientist C.B. Van Neel. C.B. Van Neel. He has conducted bacterial photosynthesis. He has conducted bacterial photosynthesis. He has conducted bacterial photosynthesis and said that C.B. Van Neel has conducted bacterial photosynthesis. And he said that oxygen molecule whichever is liberated at the end of photosynthesis is from water molecule but not from carbon dioxide. Okay, it is from water molecule. So, these are the most important and prominent discoveries that you have to know about. Fine, right. Most important discoveries. Now we will see each discovery in detail. Discoveries of photosynthesis. Just now we have learned. Now we will see first that carbon dioxide is essential for photosynthesis, which is said by Mole's half-leaf experiment. Mole's half-leaf experiment. Fine, right. Where a healthy plant which is exposed to sunlight is taken, a potted plant, and half of the leaf is immersed into a beaker containing potassium hydroxide solution. Okay, this contains potassium hydroxide solution. Okay, and half of the leaf is exposed towards the sunlight. Half leaf is immersed into the beaker and half leaf is exposed to sunlight. This potassium hydroxide solution whichever is present inside the beaker, it will react with carbon dioxide whichever is present inside the leaf and this turns to potassium carbonate. Okay, and this turns to potassium carbonate. Here when this leaf which is half exposed to sunlight and half exposed to the beaker containing potassium hydroxide, After some time, when this leaf is being conducted with starch test, it is shown that half of the leaf which is exposed to sunlight has turned to blue black color, and half of the leaf which is kept in the beaker, it shows pale color, indicating the absence of starch. This indicates the absence of starch. Fine, right? Clear? So this says that carbon dioxide, as starch is not formed in the absence of carbon dioxide, this says that only in the presence of carbon dioxide starch takes place, starch forms. Fine, right? This indicates only in the presence of carbon dioxide photosynthesis occurs. And then if you see next about oxygen discovery. Oxygen, which is discovered by Joseph Priestley in the year 1774, where Joseph Priestley has discovered oxygen. Fine, right? Oxygen discovery by Joseph Priestley in the year 1774. Fine. So this experiments, bell jar experiment, has started from 1770. Series of experiments started from 1770. Okay, bell jar experiment. What does this experiment actually says? First, inside a bell jar, a live mouse and a lightened candle is placed. After a certain period of time, it is observed that the mice dies and the candle is put off. When the same experiment is repeated with a mint plant kept inside the bell jar, the reaction is as it is. Mint plant is healthy, mouse is alive, and candle is still lightning. With this result, Joseph Priestley has said that there is some gaseous exchange that is happening between the live mint plant and the mouse candle. Okay, the gas which is released or exhaled from the mint plant is being taken by mouse and candle, and the gas which is exhaled from the mouse is being taken by the plant. That gaseous exchange is reported by Joseph Priestley. The result has made him to prove or discover that oxygen is discovered in the year 1774. The result which is seen here made him to discover the oxygen in the year 1774. Fine, right? And then it is Jan Ingenhouse's experiment who has proved that sunlight and water are essential for photosynthesis. Okay, Jan Ingenhouse. Who said sunlight and water are essential for the process of photosynthesis. So this plant, the plant which is taken here is hydrilla. Look at the experiment carefully. First, a beaker containing water is taken. Inside which the hydrilla plant is kept into a testil funnel. A testil funnel is taken into which a hydrilla plant is kept. That testil funnel into which the hydrilla plant is kept is inverted inside the beaker containing water, and above the testil funnel a test tube is kept as such. Okay. After certain period of time, when the setup is kept under the sunlight, there are found some bubbles which are found inside the test tube. When the same setup is kept in a dark room, there are no bubbles identified in the test tube. When the gas is checked, like which gas is being formed inside the test tube, when the lightened candle or a matchstick is kept over the test tube, it will burn vigorously, indicating the presence of oxygen. As such, he proved that oxygen is liberated at the end of photosynthesis. But he didn't say like oxygen is being released from carbon dioxide or water molecule is not said by Jan Ingenhousz. Okay. Now, sunlight is essential for the process of photosynthesis is explained by him. But what spectra of sunlight is essential for photosynthesis? Nobody explained. Okay. What are the active regions of the sunlight? What is the maximum wavelength of sunlight which is required for photosynthesis is not explained by Jan Ingenhousz. So, it is the scientist E.W. Engelmann who has done elegant experiment using the model organism Cladophora, which is the filamentous green algae. He discovered what? Action spectra and absorption spectra. Clear? Fine, right? Enough. So, both action spectra and absorption spectra are clear to you. Now, if we move on to photosynthetic pigments, what are the photosynthetic pigments? Most leaves will appear green as they reflect green color. Okay. The color which what is that being absorbed? Blue and maximum blue and red colors are being absorbed from the WebGR, which is proved from Engelmann's experiment. Here what is the model organism, what is the procedure of the experiment, you have to be clear with it. And some leaves are of different colors due to the presence of different pigments. The pigments can be carotenoids and xanthophylls. Amongst the constituents of white light, red and blue wavelengths are the best to carry out the photosynthesis. Okay, red and blue lights are best to carry out photosynthesis. This is what proved by Engelmann experiment. And now we will look on to what is action spectra and absorption spectra. So, two spectrums which are given in our text, very important, we can get a graph based question also. First, what is absorption spectra? And second, action spectra. First, absorption spectra, if you observe, different pigments absorb light at different wavelengths is being seen in the absorption spectra. Maximum amount of photosynthesis or maximum oxygen evolved, or maximum amount of photosynthesis is conducted by a single pigment that is shown in what? Action spectra. Okay. Diverse pigments absorbing diverse wavelengths of light is depicted in what? Absorption spectra. So, first graph will show you what is absorption spectra. Absorption spectra. Here if you can see, first let us go with chlorophyll A. Chlorophyll A will show you two peaks, one in between 400 to 500 nanometers and one in between 650 to 700 nanometers. Two peaks are observed, fine, right, of chlorophyll A. Chlorophyll B, one largest peak in between 400 to 500 and one very small peak is observed in between 600 to 700 nanometers of light. Okay. And then carotenoids will show two peaks, two equal peaks in between 400 to 500 nanometers itself. Here you can name out 400, 500 nanometers, 600 and 700 nanometers of wavelength. So, wavelength is being taken in the x axis and absorbance of light is being taken in the y axis. Remember what is being plotted over x and y axis carefully, and where the peaks are being shown by different pigments like chlorophyll A, B and carotenoids. It is important. right? And then if you see action spectra, okay, action spectra. Action spectra. Here the rate of photosynthesis release, this is measured by oxygen release. So here a single pigment will show two peaks, one in between 400 to 500 nanometers, and the equal length highest peak is observed in between 600 to 700 as well. So two peaks are observed by chlorophyll A. Now here in the third graph you can observe the rate of photosynthesis and absorption of sunlight, okay. Both absorption spectra and action spectra of chlorophyll A will resemble each other. See here, if you see maximum absorption by chlorophyll A, that is in the blue region and the red regions, both in blue and red regions, maximum absorption of chlorophyll A is observed with higher rate of photosynthesis. Hence we can conclude that chlorophyll A is the chief pigment associated with photosynthesis, and chlorophyll A absorption spectra will coincide with that of action spectra, okay. That is a major statement that is to be highlighted. Now where is the photosynthesis happening? I hope the complete discoveries part is clear to you, right? I hope the complete discoveries part is clear to you, right? Once again, let us take all the discoveries in a single slide, because from discoveries the questions may be put in the match the following type of question. So be clear with the discoveries of photosynthesis. Look at here, which is the standard equation of photosynthesis. Fine. Now, carbon dioxide is essential is said by Mole's half-leaf experiment. Second, sunlight are essential is said by Jan Ingenhouse. Fine. Glucose is the product of photosynthesis. This is being said by Julius von Sachs. Fine, right, ma. Now when it comes to oxygen, three major discoveries you have to know. One, first one, oxygen discovery by Joseph Priestley. Bell jar experiment. Fine. Second, oxygen evolved from water. This is said by what? C.B. Van Niel by conducting bacterial photosynthesis. Okay. Here in bacterial photosynthesis, as it is anoxygenic photosynthesis, H2S will be the electron donor, where it will be releasing out sulfur. In the same way, in oxygenic photosynthesis, water will be the electron donor, where it will be releasing out the oxygen. And third discovery is by Jan Ingenhouse, which is third discovery. What is that? Oxygen is released at the end of photosynthesis. If you can remember this chart, complete discoveries are covered. Okay, complete discoveries are covered in a single slide. You can also add on action spectra and absorption spectra. You can add on here. Action spectra and absorption spectra. Action spectra and absorption spectra are given by whom? E.W. Engelmann. Fine, right. Clear with the complete discoveries? Any doubt anywhere, ma? Now, we'll move on to the locations or sites before which we have to completely know about chloroplast organelle. Chloroplast organelle. Of course, it is double membrane bound organelle. The space between outer and inner membrane is known as periplastidal space. Space between outer and inner membrane of the chloroplast is known as periplastidal space. And the third membrane system which is present inside the chloroplast organelle, sac-like structures, membranous sac-like structures are known as thylakoids. Stalk of this sac-like structures are known as grana. One grana is connected to other grana by stroma lamellae. By what? Stroma lamellae. And the fluid filled matrix inside the chloroplast is known as stroma. What is stroma? Fluid filled matrix inside the chloroplast is known as stroma. Inside stroma, what are present? Lipid droplets, starch granules and ribosomes. Ribosomes are of 70S type. 70S type of ribosomes, whose larger subunit is 50S and smaller subunit will be 30S. Okay, these are 70S ribosomes. Clear till here? Any doubt anywhere? And chloroplast is also addressed as semi-autonomous cell organelle. Okay, this is semi-autonomous cell organelle. It consists of a double-stranded circular DNA. Double-stranded circular DNA. Double-stranded circular DNA. Clear till here? Any doubt anywhere, ma? Now, we will look into the events that happen in the photosynthesis. What are light dependent reactions? What are light independent reactions? Let us look into it clearly. First, photosynthesis, both light reactions and dark reactions. First, light dependent reactions. Second is light independent reactions. Calling light independent reactions as dark reactions is a misnomer. Okay. Here we go with light dependent reactions. What are the three major events that occur inside the light dependent reactions? First is absorption of light. Absorption of light in the form of photons. Fine. Second reaction is photolysis of water. Photolysis of water, where water molecule is photolysed to 2 protons, 2 electrons and an oxygen atom. Fine, right? And third is formation of assimilatory power. Assimilatory power. Assimilatory power is ATP and NADPH. ATP and NADPH. What are the events that occur in light independent reactions? Only thing that is formation of glucose. Formation of glucose. Now let us clearly monitor the sites or locations of the reactions. Here we look into first, absorption of light in the form of photons occurs inside thylakoid lamellae. Okay, this occurs inside thylakoid lamellae. Photolysis of water happens in oxygen evolving complex, which is on the inner side of thylakoid lamellae towards the lumen. Okay, inner side of thylakoid lamellae towards lumen. Fine. Formation of assimilatory power, that is ATP and NADPH. This occurs inside stroma. And glucose formation also occurs inside stroma. By what cycles glucose formation occurs? Okay, C3 cycle is the common cycle that operates in all the plants. Okay, first is C3 cycle operates in all the plants. Next through C4 cycle in tropical plants. Next is CAM cycle, which is operated in desert plants. These are the locations or sites of the reactions. Clear till here? Any doubt anywhere? Take down the screenshot of this. This is highly useful for you people. About the locations or sites of the reactions, many at times the questions are asked. Fine, right? Now, look at the light harvesting complex. Okay. So, we have said that the very first step of light reaction is absorption of light in the form of photons. Where is light absorbed in the form of photons? This is being absorbed in light harvesting complexes. Okay. Light harvesting complexes. Where are these light harvesting complex present? Let us suppose this as the chloroplast organelle and this is the thylakoid membrane. And we have discussed thylakoid membrane, it has thylakoid lamellae, which is thylakoid membrane, and inside which lumen is present, right ma? So now let us suppose this is the thylakoid membrane, this is lumen and this is thylakoid lamellae. Fine, right. Inside the thylakoid lamellae, light harvesting complexes are present. In oxygenic photosynthesis, you can see two light harvesting complexes which are addressed as photosystems. Fine, right, photosystems. As such, two photosystems are present inside the thylakoid lamellae. Here you can see photosystem 2, which is PS2, and then photosystem 1, that is PS1. Photosystem 2 and photosystem 1. Inside each photosystem, you have two types of pigments. Okay, two types of pigments are present. Okay, one is core pigment and second is accessory or antenna pigments. Accessory or antenna pigments. Core pigment will be chlorophyll A. Okay, chlorophyll A. Accessory or antenna pigments will be xanthophylls. First is chlorophyll B, xanthophylls, and carotenoids. Chlorophyll B, xanthophylls and carotenoids. Accessory or antenna pigments will absorb light at varied wavelengths. At diverse wavelengths, light will be absorbed by accessory or antenna pigments. The light which is absorbed is being transmitted to the reaction center. Okay, light which is absorbed by accessory or antenna pigments will be excited and transmitted to the core pigment or the reaction center. That reaction center is chlorophyll A. The maximum wavelength of chlorophyll A will be variable in both the photosystems. In PS2, maximum wavelength of chlorophyll A will be P680. 680 nanometers is the maximum wavelength of the reaction center. Here in photosystem 1, chlorophyll A will have maximum wavelength, that is P700. This is the maximum wavelength of chlorophyll B. In our next class, we will be looking into the photophosphorylation, which is the major concept, cyclic and non-cyclic photophosphorylation, photolysis of water, C3 cycle, photorespiration, that is C2 cycle, CAM cycle and C4 cycle, which is the most important topics along with bioenergetics we will be dealing in the next class. Okay, so that's all for today's session, students. Thank you. If you are having any doubts, please post in the comment box. I will be addressing all your doubts. Okay, thank you.